Hi, good morning, and welcome to the ZP Developer Zone. So we like to do this Developer Zone a couple of days a week because it's been getting very busy with questions. So I'm going to dive straight into it because there's lots of questions and we want to get through them. I also want to apologize slightly if there's a bit of a background noise. I do have a good microphone, but at the same time I am running a 3D printing in the background. So I apologize for any background noise. Trying to do this, answer questions for people who've asked questions, and also trying to do some 3D printing as well. But today's the 4th of February, 2023. A lot of what we do now at Zimmer Peacock is really about the Sensi All platform. The Sensi All platform is most matured in terms of IVD, in vitro diagnostics. So somebody's asking about lateral flow today, or the combination of electrochemistry and lateral flow, and this platform is actually ideal for them. And we also have this kind of platform also available for the wearables as well, and wearables will come up quite a bit today as well. That said, let me go forward, and I suppose the Sensi All platform really kind of reflects the whole philosophy at ZP, which is, and you'll see it today a lot as well, as actually how to get people out of the lab and onto the market. I mean, that's always been the philosophy at ZP, and remains so today. By the way, we are ISO 13485, so we are contract developers, contract manufacturers of electrochemical biosensors. That said, there is a webinar on the 27th of February. If you see a link jump up today, those links, I'll put them underneath the video. Please sign up, especially if you're interested in lateral flow, because I think we should be covering the Sensi All platform on the 27th of February. So we do one in the morning and one in the afternoon. That's London time. The reason being is the one for the morning is for people in sort of one side of the globe, and then we do one in the afternoon for the other, essentially the other half of the globe. So same webinar, but they're live and there's live demos. Also, if you're a large corporation and you want to kind of engage and you really want to get some kind of learning around biosensors, ZP does do on-site corporate workshops. We also do workshops on our site. So there's at least there's about 2 or 3 questions today that have come on CGM, continuous glucose monitoring, and then there's a few questions around IVD as well. Just know that we have these workshops at our site. So there's 2 types of workshops. There's at your site and there's at our site. But on the I think it's the 5th and 6th of March, it's a 2-day workshop. It's quite comprehensive. It's very hands-on, and a lot of the questions that have come in today are actually all around CGM, continuous glucose monitoring. So please come to those workshops. If you're serious and you have serious questions, then come to those workshops. And I'll put a link underneath the video as well. As I say, we do go on site and we do workshops for people, but you have to be able to get together 12 to 20 people in order to make that kind of useful. We're also having a conference in the UK in about June-ish, I think, and I'll also put a link there. Now I'm going to get to the questions. So that was a bit of a spiel. Now I'm going to talk to questions. Right now, question number 1 is about electrochemical lateral flow strips. It's probably worth saying that I did cover this about a week ago, but they came back with some more questions, and I'm going to answer those questions today. ZP, we have a lot of technologies that are very advanced when it comes to CGM. I will tell you what we've got, and then it's for you to judge whether these are interesting or not to you. Somebody else is actually, on our website, we do have some reagents to allow people to make CGM. The thing is that if you the reagents are very well described on the website. There are videos there on the website. If you can follow the videos, then that means that you are fairly qualified to actually do the process. If it's not clear to you from the videos what to do, then you will have to engage us in consulting, because paid consulting, because the videos are kind of like a barrier. Like, if you can follow the videos, then you are probably qualified to be able to do that kind of work. If you're not, then you need guidance, and therefore you'll need some consulting from Zimmer Peacock. But I'll cover that today as well. Somebody else was asking about what kind of lab performance do they do in order to kind of think that they've got something that's good enough to go to market. Now, this particular person is kind of modifying things like screen printed electrodes. I think they're actually modifying actually, when I think about it, something called a glassy carbon electrode. And I'll cover that today. It's just coming back to me what they're talking about. So I'll talk about lab performance and how that could then translate into real world performance. Somebody else, East Coast USA, is interested in our glucose, lactate, and a platinum electrode. And they're interested in SPE connectors, screen printed electrode connectors. So I will discuss that today as well. what I will say is that ZP will support these sensors and electrodes when you're using our electronics. It's either our potentiostats or our single purpose biosensor circuits. I do have a lot of information on YouTube and on the website about various biosensors. If you're able to follow that, that's great. And if you're using third-party potentiostats and you can follow what we've done on our website, great. But I can't support third-party potentiostats with our stuff because then I'm trying to troubleshoot that potentiostat that I haven't otherwise supplied versus our sensors and screen printed electrodes. So, but most people just can, if they've got experience, they can just get on with our sensors on their own without that much support from us. That said, question number 1, screen printed lateral flow strips. Let me jump into it. So at ZP, we do make a really repeatable screen printed electrode. This is really important. Otherwise, you can't make IVD in vitro diagnostic type products. And even if you're making microflow cells, you do need a good repeatable electrode at least. Otherwise, you've got a variation in your signal that's due to the electrode itself. But when we are at ZP, I have been interested in the integration between lateral flow and electrochemical screen printed electrodes for quite some time. Because I feel like, lateral flow is a good technology. It's scalable. It does kind of work. COVID-19 wasn't perfect, because if the line's a little bit faint, it's hard for people to be able to interpret it. But I like the integration between electrochemical sensors and lateral flow strips, because you can kind of get the separation on the lateral flow strip and then the sort of digitization of that signal on the electrochemical sensor. So I do like the idea of integrating these two things together. I don't think anyone's actually done it commercially, but there is a lot of similarity between a glucose biosensor, or sorry, I should just say electrochemical biosensor, and a lateral flow strip, that you have a sample going in one end and the sample kind of flows into these kind of devices. By the way, this is a what I call a capillary flow sensor that we do make at ZP, and we do it commercially for people. But I do see the similarity between lateral flow and biosensors. Also, if somebody is making a lateral flow device, if they kind of follow the kind of guidelines that I'm trying to lay out today, they'll actually be able to use it on this Sensi-All platform. So that really puts them a lot closer to a product than they would otherwise imagine themselves. So we're really good at ZP of kind of seeing what people want to achieve and then making it work on the platforms that we have, because then they can actually get to market much quicker than they would otherwise have imagined, let's say. So there's a couple of ways of integrating a screen printed electrode with a lateral flow strip. There's this kind of orthogonal way on the left hand side, or there's this kind of linear way that I've shown on the right hand side. I mean, I said it last week and I'll try to give a bit more detail this week. I don't like this design. I mean, there's I suppose I've got a device here. This is our Sensi-All platform. It looks nice. It's sort of something that, you showed it to a doctor or an end user, they would kind of understand it. But when you show them something that looks a bit kludgy like that, it's a bit jolting, to be honest with you. So, I mean, I know it sounds ridiculous, but there's an aesthetic to this that's not easy to, it's not an easy to, the device is not particularly pleasant to kind of look at, and it actually doesn't, I don't think it would actually handle very well either. Whereas I quite like this design, but I will talk about my kind of prejudices against this orthogonal design shortly. I like the linear design. It would be lovely if you could put the lateral flow material on top of the screen printed electrode, put your sample on, put your buffer on, and then it would just flow and detect. I suspect that some of the distances here are a bit short. And at ZP, I'll say it a few times, we're ISO 13485, which means that we do risk assessments on hardware before we actually start, maybe going too far down the R&D route. And here I do a risk assessment and say, I'm putting that liquid a little bit close to where I'm actually want to make an electrical contact. So obviously liquid and electricity never mix well. So I don't like the idea of having to put liquid here. It's there, I'm a few millimeters away from that connection. So that's a risk, because you'll end up, somebody, especially real people in the real world, will end up splashing material onto those electrodes, and then it'll be shorting when they actually try to do the measurement. So even though I've put it up there, I'm just saying, you've got to do a risk assessment on this. This, I must admit, looks a bit kludgy, but I do like, this with this design, you put your sample on, you put your buffer on, and it flows down. The nice thing about this design is you can make this kind of length anything you want. And I should have said it, if you were actually making a product, you then didn't need to necessarily have this length particularly long. So this linear format I do like. a lot more. And I really don't like this orthogonal format at all. It's what's been happening in the literature. In the literature, people are really doing kind of proof of principle, lash it together. Does it work? Yes, it does. Publish a paper, done. But it doesn't really kind of think about the kind of whole customer experience. So I do like the middle one, and I certainly don't like the other. Well, I quite like the far left one. It's just I don't think it's good to add liquids quite so close to the electrical connection. So that has to be thought through a little bit more. I don't like the orthogonal design. As I say, we are ISO 13485, which means that as part of doing this kind of contract development, contract manufacturing, we actually do a risk assessment on this, and I could sort of see this being a bit of a risk. My risk assessment, let's say, on it starts with this, that you have this kind of orthogonal design with a lateral flow strip, flowing, running in one direction, and then orthogonal to that you have this screen printed electrode. That means you've got quite a length here. So length can mean quite a leverage. A little bit of movement here can put quite a bit of strain at the pivot point down here. So I feel like this length is actually a bit of a problem, because any small movement any forces here will get sort of amplified along this length. These connections that you see people using, they can be a little bit flimsy. So I can easily see in the real world, if I was thinking about a nurse or a patient or a doctor, they will knock this, and it will interrupt the electrical connection here. So this is kind of dead in the water before it hits the clinic. Yeah, you can publish a paper on it, but honestly, as a risk assessment, I think there's a bit of a problem here. This is more of a kind of commercial problem, that you've got something with a length and a width. Therefore, you have to package it in quite a large package. And I know one thing about this kind of technology. People will always be arguing, oh, I want low cost. Well, low cost in part comes from size. So with here, you've got it's a lot of dead area, but you still have to package it in something that's fairly big. And packaging is costly in its own material rights, but it's also costly in the logistics, that you can't pack so many in a box. You can't pack so many of those on a crate. It really does multiply the problem. So that width and that length, when you've got the orthogonal design, I think it's a bit of a problem. So I kind of think it's awkward to get into the connector. So you're going to push that guy into the connector, having this guy sort of lying along it. That's kind of awkward. It puts a lot of leverage on the connector. People will knock it. It's awkward to package. You kind of design something that's, you're not using all that area, but it's actually fairly big because of the length here and the length here, and you'll be surprised at how quickly the packaging costs. I think it makes it expensive to sort of handle and do, and I don't think there's it's really linked to the two bits above, awkward and leverage. It's not a good customer experience. So I know it's a bit, in fact, I'll just drag myself slightly out of that. So I'm just giving you almost like the sort of the vision towards the horizon, because in the end at ZP, the whole philosophy is everyone should get to market. And some things that would otherwise happen in the academic world would have to be rethought out before they get to the real kind of end user. They did have some specific questions. I mean, this is elegant, but the problem I've got with this is I suspect that there's not enough length along this track in order to get the separation before you get the detection. And I also, as I say, I don't like the fact that maybe that liquid is a little bit close to that screen printed electrode. This can possibly be improved. I do like this, because you can have all the length you want in the world. It does look a bit kludgy the way I've drawn it, but I do in fact I like it's linear, because when it's linear like that, you can basically put them into tubes. You can, if you look at a glucose strip, glucose strips are shipped in cylinders. So in cylinders, they can put like 50 of these sensors in a cylinder. So I can see the cylinder having to be longer, but I can see you being able to just pop all these into a cylinder, and it'll make for a much denser, let's say, packing. And again, I don't like that, and I have sort of given my reasons why. The particular inquirer is interested in knowing the length along here, because I can see that they're thinking about this. I think it's okay to do a paper on this, because if you can prove that it works, then you can think about the next design. So I think the first step is get away from that orthogonal design, try and get towards a linear design. The length along here is what you were asking about. It's 17.2 millimeters. I'll just give you a better drawing here to sort of show you how I got to that point, because I can see the length from the reference electrode here to the electrical connections here is about 19.2. I can see these electrodes are kind of 1 millimeter wide, so I just kind of did a quick estimation of that, and it's about 17.2 millimeters. I do understand their question, which is, will I have enough length in order to get enough separation? Quick answer is you could always try it. One thing I'd say about science is that it's good to do your calculations, and having done your calculations, then you can kind of confirm it with a quick experiment or an experiment, and then it's all publishable, because you kind of your theory and even if it didn't work, your theory and your practice kind of do tie up with one another. So I just say give it a go, to be honest with you. If they can get with this linear design, then they can actually get with they can get onto one of our platforms, which is in fact this platform. This is what we're going to have the webinar on the I think it was the 27th of February. Come along to that, because then you can actually get onto this platform, and suddenly you're in a much better place when it comes to commercializing this idea. So just to back up, they need to make this kind of combined lateral flow screen printed electrode. The nice thing about using our screen printed electrodes, especially our 7 millimeter diameter electrodes, is that they're compatible with this platform. We can do QR codes that will tell the hardware what to do. The hardware works with an app. I don't have it today, but there's apps on the Play Store and on the Apple Store as well. And then they get a really nice experience, because you can get a result on the phone, but also you can get the raw data into the cloud as well, which would be really important for these people as they're trying to develop and troubleshoot. And then they can share with people like us, or share it with their own colleagues. This is a bit kludgy, but I'm just showing you that it's not that hard for us to be able to screen print, not screen print, sorry, it's 3D print, which is ironic, because I've got a 3D printer running behind me at the moment. That's why I'm shouting slightly, to try and get rid of that horrible white noise that's going on in the background. But anyway, we are able to 3D print adapters that fit in the Sense It All. So I could sort of see myself with an adapter that had a lateral flow material on it as well. So for these people, I'm recommending a particular electrode, this hypervalue carbon electrode. I know that you're considering it, but I also know it's a challenge for you, and I know that you would like, and I'll put some links here. There's a particular electrode they do want off us, and I would ask these people to come to the 27th of February webinar. Again, if the link's underneath here, I'll put it underneath the video. Just a quick apology to these people, because they really want this electrode. I don't like it because of the orthogonal sort of design that they're considering. But that said, gold has become very expensive. If anyone's trying to make it in low-cost biosensor at the moment, really, if you can, avoid gold, because in fact the reason we're doing this in quite large pack sizes, and we can't do it in smaller pack sizes because gold is intrinsically quite expensive, so we have to be really efficient with our use of the gold. So we just can't do small pack sizes. I'm sorry, but people will, there are gold electrodes out there. Gold has become super expensive. It's not necessarily a good material anymore for making in vitro diagnostics. In economic times like this, a lot of people go to gold in terms to put their money, so suddenly there's a much more of a pressure on gold. So we can't do smaller pack sizes is the quick answer. Somebody's asking whether we have a CGM that's ready for manufacturing. It's probably worth saying that we are already manufacturing CGMs. At ZP, we do have a lot of experience in transdermal CGMs. So this is the kind of wire configuration that we have. This is the microneedle configuration that we have. This is, I'm not going to suggest sweat for people doing continuous glucose monitoring. There is a company doing it. I'm sure they've got some IP around it, and it's going to be a very hard piece of clinical work to prove that glucose in sweat is representative of glucose in interstitial fluid. I think that's a 20-year journey, so I'm not going to recommend that in any way. We do have implanted sensors that are CGM as well. I'll show you a bit more of this. I'm probably going to focus on the microneedle, the implanted, and the wire this morning as well. This is actually a system that gets implanted. You can see the maturity in this. This has had several million worth of euros spent on it. It's primarily used these days. It sounds ridiculous, but it's primarily put into fish, though it's also getting some mammalian applications as well. These are the fish swimming around with these completely implanted wireless CGM systems, and this is the glucose data that's kind of coming off those fish. This is something that we also have at ZP. This is, oh, in fact, I've got some here. I'm sorry about the rustling of packages. I just happen to have a couple of here, actually. So my apologies for the rustling. Maybe I'll just go forward slightly. Yeah, that's a better picture. This is a minimally invasive CGM, continuous glucose monitor. It has micro needles on the back side of it. This is the patch itself. This is the watch. The micro needles go on the back of the watch, and the watch goes onto the wrist. A couple of more configurations. This sort of shows it a little bit. When you pull on the when you pull on the strap, it pulls the micro needles through the skin. So this is something that's this has had something like 20 million euros spent on it so far. Ridiculous, not ridiculous. I mean, this is what it costs to make a CGM. And in fact, it's protected by approximately 50 patents. I have to say approximately 50 patents. That is something that we are licensing out to people, so that is available. But I'll be straight, it really has to come from, it can't be for a small high school or university project or a startup that isn't well funded. But we do have some really good CGM technologies, but we would have to do some due diligence on the company, asking who are you, what's your company, what's your kind of investor profile look like, because it's a serious bit of technology, and we would really need to we'd have to do some due diligence before entering into that conversation. But please contact us if you are so interested. This is another question where somebody wants to make their own CGM, I'm going to sort of say here that if the videos are not clear enough on the website, then I suspect that you'll probably need us to consult into you, so you will have to get a consulting program with us. But if people want to make their own CGM, we do make some materials available on the website. It's probably worth saying that we just do it for R&D purposes only. It's the same for, like, if you go to Sigma. So Sigma is a big chemical supplier, and a lot of people buy their materials. If you read their terms and conditions, they'll actually say for R&D purposes only. Most people have never read their terms and conditions, so don't realize this, but it does actually say it there. So people can buy our materials, they can try them out, they can make their own CGM, but it's only for R&D purposes only. And if you want to have a commercial conversation with us, then you have to get into a paid program with us about that. If the videos are not clear enough, then I would suggest that it probably means that, you know, your team or you are not quite ready to actually do this kind of work, and you probably need some consulting off us, and therefore you can get some consulting support off us at ZP. But I suppose it does say that we do have a lot of CGM, continuous glucose monitoring know-how at ZP. I mean, this is one of those other glucose watches here. This one's a kind of more of a sort of sports configuration, but you can see that we have technologies at so many levels: system level, materials level, electronics level. It's just a way background. I've been making glucose strips since 2000, so it's been going on quite a long time. Of course, this is about CGM, continuous glucose monitoring. Question number 4. I've been not advising, but I've been on and off questions and answers with this person for a while rather. They're trying to make a non-enzymatic small molecule sensor, and I remember asking them, actually, are you doing it on a screen printed electrode? And they're not. They're doing it on glassy carbon electrodes. So if you're still on glassy carbon electrodes, you're not. You have to get onto something like a screen printed electrode, because you're not moving forward to the market. You're just really in a sort of R&D phase. If we were talking about glucose sensors, you know, this is actually a ZP. We not only have the technology, but we also have entire sort of software systems based up for analyzing CGM, continuous glucose monitoring. So this is the data from a couple of CGMs that we're testing in the lab. These are prototypes or R&D pieces. But we can go in there and I can kind of look at the raw signal there, and I can ask myself the question, what is that raw signal? That signal there is 1.71 microamps or 1.48 microamps. So if I do the average of those two, I get 1.59, and if I do the standard deviation, I get 0.16. So you don't have to be much of a maths genius to see that my relative standard deviation, that's my standard deviation divided by my average, is about, and times by 100, is about 10%. Realistically, if you're in the lab and you're on a glassy carbon electrode and you test with a known concentration, let's say I had, you were testing a glucose solution and it was 5 millimolar, you should test 5 sensors, let's say, and you should get a signal because it's the same solution, and you should get a signal when you do the relative standard deviation for it that's less than 5%. That's in lab conditions. Then you're sort of ready to go ahead. Yours is a bit tricky because you're on a screen on a glassy carbon electrode. I think you're very far from actually really even asking this question, because the most important thing when commercialization is not necessarily cost is important in commercialization. Cost is linked to materials and linked to ease of manufacturing. But then it's actually repeatability, which is important, especially in IVD and in vitro diagnostics work. The reason it's more important in IVD because you put the sample on there, you get a result, you're done. You've got very little time for dealing with kind of calibration routines, etc. So you need a really good lab repeatability to even stand a chance in of doing this commercially. Question number 5, somebody's interested in glucose, lactate, and potassium. We do have, all these formulations, glucose, potassium, lactate is here. They're interested also in a screen printed electrode. In particular, they're interested in platinum. These are the electrodes that I kind of most recommend to people. These are all 7mm in width. These are the same electrodes that I'm recommending to the person who was trying to make lateral flow technologies earlier on. This particular person is interested in platinum. This is an easy electrode to use because it's got a circular electrode. So that 501 platinum is what I recommend. Some links are going to jump up now. There's also this hypervalue platinum. Perfectly good electrode. I'm just sort of thinking in fact, this person's thinking about making a flow cell. So that is a little bit of I might have changed my opinion as I think about a flow cell. But in order to get you going, well, you could try both. But 501 would be slightly more over the hypervalue. Because of your flow cell, I was just thinking, oh yeah, actually, you might want a linear electrode. But I don't know your flow cell. I think you'd have to try both of them, see which one gave you the best signal. I suspect they would both work, and the 501 is easier to pipette onto. So when people are in a laboratory, I think circular electrodes are better. We have all the robots, so we don't care about what the configuration of the electrode is. But that said, there's a 501 platinum link here. I hope you can follow that and make sure. You're also asking about connectors. The reason I wanted to do this in a video is because I want you to know that the glucose, the potassium, and that screen printed electrode, they're all 7mm width. So you need a 7mm connector. And I don't know your potentiostat. I'm suspecting that it's got either 2mm or 4mm diameter banana plugs. You have to check, because then we'd either be supplying you the 2mm banana connector or the 4mm. But that's the bit that I need you to check, because then I can advise you which connector. We will support our sensors if people are using, for example, calibration solutions that have come from us. There's nothing worse than trying to help people with their own electronics and their own solutions, because their own electronics and their own solutions are outside of our control. The things that we can control are things that we've manufactured ourselves. So the more parameters that you give us that are outside of our control, for example, you in the lab is outside of our control. So there are, in life, there's things that you can control and things that you can't control. So when people try to kind of get us to help them on electronics that we haven't supplied, that is a lot tougher. But just saying that, you didn't ask us for electronics, but I just want to let you know that we do have these single-purpose biosensor circuits. So if you have an electronics background, only if you have an electronics background, this is an option. We also have our potentiostat. This is a 6-channel instrument. We use it a lot, and you can use that with our biosensors as well. And then I prefer you using that because obviously I'm biased that I'm in the video. It's got ZP on it, and it also means we can support it because we understand that it does work. Whereas I don't like troubleshooting other people's potentiostats. Recommendation, I think in the short term you might just want a glucose sensor off us, so that will be the glucose sensor that I recommend. I'll put the link underneath the video. You can check it, make sure it's okay. And then it's either a 7mm 2mm connector or a 7mm 4mm connector, depending on what your potentiostat actually has. So question number 1, electrochemical lateral flow. I did give you that dimension, it was 17.2mm. I still think you should be going for a linear flow rather than orthogonal flow. The cost of gold means that it's very hard for us to actually do any pack sizes smaller than 3000. I apologize, but that's really the situation. In terms of the other person, we do have some very serious technologies that have had 20 million euros invested in them. But I need to know who you are, what your company is, are you well funded, what's your business ideas, etc. Because there's a bit of we'd have to do due diligence back on you. Making a CGM. There is a website, there is a web page, there are 2 videos. You can buy the solutions, you can pipette them on, you can make your own CGM on your own substrate. If you get into problems or can't understand that, then you probably need consulting from ZP. Repeatability, you should be able to get repeatability when testing the same solution on different electrodes or sensors of less than 5%. I think it's very hard to do it in an R&D in a lab environment in an academia, but you asked and I gave you the answer. Here I've suggested a Gen 1 glucose sensor. I'll put a link underneath the video, and I've suggested the 2 screen printed electrodes. This person's at a very prestigious universities, and I think they're also quite experienced, so I think they should be fine. But if you're watching this video and you are not so sure, then you might need consulting from Zimmer and Peacock. So I shall wrap up with saying thank you. This is the Contact Us. There will be another webinar on Thursday. We get a lot in these days. You can see we put a lot of effort into making these webinars. And if you've got any questions, please don't hesitate to reach out to us. Okay, thanks very much.